Howdy! In this mini lecture we will discuss environmental toxicology, dose-response curves, and types of toxicants. The student learning objectives of this mini lecture are: distinguish between epidemiological studies and manipulative experiments as they are used in toxicology, explain the importance of wildlife toxicology and the significance of Dr. Guillette's and Dr. Hayes, interpret a dose-response curve, and correctly identify the classes of toxicants. Environmental toxicologists seek to understand the effects of toxicants and toxins on organisms in their natural environment. The efforts of these scientists can help conserve wild populations and shed light on the risks certain compounds pose to human health. Both fieldwork and lab studies are used in environmental toxicology and wildlife studies. For example, the picture on the left shows a famous wildlife toxicologist, Louis Guillette, taking blood samples from an alligator in a Florida lake. Dr. Guillette was comparing the blood of alligators that lived in lakes that received agricultural runoff to alligators that had lived in lakes that did not. He hypothesized that chemical contaminants were disrupting the endocrine system of alligators during their early development inside the egg. He later learned through lab experiments that the herbicide atrazine was the source of problems and was indeed disrupting the endocrine system of the reptiles. Building on Guillette's work, Tyrone Hayes found in lab experiments that male frogs raised in water with very low levels of atrazine became feminized and hermaphroditic. Furthermore, field studies showed frogs in areas with heavy atrazine use have hormonal problems. To study human health, scientists use epidemiological studies. To do these studies, epidemiologists compare groups of people that were exposed to a hazard to groups that were not exposed and do so on a large scale. Epidemiological studies can last for years or decades. If rates of death, cancer, etc. are higher in the exposed group, the hazard may be responsible. Epidemiological studies show correlation between a proposed hazard and an observed health effect. They do not show causation, meaning a definitive cause and effect. Manipulative experiments, on the other hand, show causation, but these are unethical in humans. Case histories are individual cases of disease in individual patients. While helpful to some degree because they educate us about human illness, they are not all that effective at helping us understand chronic effects or the probability and risk associated with specific disease. Therefore, mammals with shared evolutionary history, such as rats and mice, are used to study substances that may harm them and us. While there are ethical concerns around animal testing, such testing has saved millions of lives of humans. If animal testing is disturbing to you, take heart to know that scientists are developing ways to test chemicals without the use of a live animal, but rather with tissues grown in a lab that stimulate a live animal and with computer models. This is still a very developing subfield of toxicology, but it is on the horizon. Toxicologists use dose-response analysis to quantify a substance's toxicity. Dose-response analysis measures the strength of toxicant effects by quantifying the number of animals affected at different doses. You should know that a dose is defined as the amount of substance the test animal receives or the amount of a compound someone is exposed to. A response is the type or magnitude of negative effects caused by exposure to a dose. A dose-response curve is the dose plotted against the response on a graph. In the bottom left, you see a dose-response curve following a normal bell curve. This shows that there are some individuals that are extremely sensitive to a dose and some individuals that are very insensitive, but the vast majority of individuals will fall in between these two categories. An important measure of a toxicity test is the lethal dose 50 or the effective dose 50, LD50 or ED50. This is the amount of toxicant required to kill or affect 50% of the subjects in the study. As you can see in the graph in the upper right, dose is plotted on the x-axis and response, in terms of percent of population killed by the dose given, is on the y-axis. A higher LD or ED50 indicates low toxicity. Take a minute and look at the graph to process that and make sure you follow why this is the case. If you have any questions about this, please email and ask. Toxicologists are often interested in synergistic effects, or the effects that mixes of chemicals have. A synergistic effect means that the mix of chemicals may be more toxic than the sum of its parts, meaning that the chemicals magnify the effect of each other when combined together. For example, DDT can cause or inhibit sex reversal, depending on the presence of other chemicals. The problem is that we can't determine the impact of all mixed hazards and that they may act in such ways that cannot be predicted from the effects of each in isolation. Mixed toxicants can sum, cancel out, or multiply each other's effects. Again, if they multiply each other's effects, this is called a synergistic effect. When looking at a dose-response curve, toxicologists are also interested in the threshold dose, or the level where a response occurs. Looking on a graph, the threshold would be where the line changes direction rapidly. You can see this clearly on the graph in the upper right-hand corner of this slide. Organs can metabolize or excrete low doses of a toxicant. Sometimes a response decreases as a dose increases. This is seen in the dose-response curve in the center bottom of this slide. Here you can see an inverted U-shaped curve. In this case, the dose does not make the poison. This curve and ones like it that form either a U or a J shape are called nonlinear dose-response curves. Such curves are counterintuitive and have been a surprise to encounter in toxicology studies in the past few decades. Endocrine disruptors are a type of toxicant that display this type of dose response. We will discuss these types of compounds shortly. Let's do a short practice. Consider the dose-response curve. What is the LD50? The LD50 is about 10 units. According to the graph, at about 10 units of a dose, 50% of the test population was affected by that dose. There are several different types of toxicants. We will consider 7 in the next 3 slides. The first are carcinogens, which are compounds that cause cancer. These can be hard to identify because of the long lag time between exposure and onset of cancer. The second are mutagens, which are compounds that cause DNA mutations. These mutations can lead to cancer or they can harm sperm or egg cells and therefore offspring. The third are teratogens, which are compounds that cause birth defects to embryos and fetuses. A famous example of a teratogen is a thalidomide. This drug was given to expectant mothers in the 1950s as a sleeping pill and a treatment for nausea. Sadly, when the children of women who took this drug were born, it was quickly discovered that the drug caused birth defects and sexual dysfunction in the children. Many of the children born to mothers who took thalidomide were unable to have their own children. Neurotoxins affect the nervous system. Examples include venoms, lead, mercury, and some pesticides. For example, people living on Minamata Bay in China between 1930 and 1960 were exposed to mercury because the nearby plant dumped it into the bay. The people ate the fish in the bay and drank the water. The results of this exposure were slurred speech, loss of muscle control, and often death. Allergens are compounds which cause the immune system to overactivate and provoke an immune response when one isn't necessary. Allergens are not technically considered toxins because they only affect some people. The sixth toxin are pathway inhibitors, which are compounds which interrupt biochemical processes in organisms. An example would be rat poison. This poison causes internal hemorrhaging and stops blood clotting. The side effect of this is that while dying, the organism is very thirsty and will seek out sources of water. If you use rat poison in your house, first, be very careful you, your children, your pets, and non-target organisms are not exposed. And 2, be prepared for a smelly house if the rat stays in the house. Finally, the last toxicant type we will consider is endocrine disruptors. These compounds disrupt the endocrine system, which is the hormone system. Endocrine disrupting compounds are called EDCs for short. Hormones are chemical messengers that stimulate growth, development, and sexual maturity. Hormones regulate brain function, appetite, sex drive, and many other body processes. Synthetic chemicals can block or mimic hormones. Bisphenol A and phthalates are 2 examples of endocrine disruptors. These chemicals can block or mimic the normal hormone signal, thereby disrupting the cascade of effects hormones naturally cause in the body. Because one hormone can have multiple effects in the body, EDCs can cause multiple effects as well. They can be carcinogenic and mutagenic as well. Endocrine disruptors pose challenges because they have unconventional dose-response curves, and these compounds are hard to study because they cause effects at extremely low doses, and it's suspected that they have chronic effects. Therefore, it's hard to set safety standards for these substances. What toxicologists have learned about EDCs means that we may have underestimated effects of other chemicals because other chemicals may also have low-dose effects. In 1996, Theo Colburn established and published Our Stolen Future. This book did the same thing for public awareness of endocrine disruptors that Silent Spring did for pesticides. Thousands of studies have linked hundreds of chemicals to effects on reproduction, development, immune function, brain and nervous system function, and other hormone-driven processes. Some researchers say EDCs may be responsible for rise of testicular cancer, undescended testes, male genital birth effects, rise in breast cancer, and other processes influenced by endocrine systems. Bisphenol A, or BPA, is a classic example of an endocrine disruptor. It acts as an endocrine disruptor by mimicking estrogen in mammalian bodies. This compound is found in hundreds of products. It is in the resins that line food and beverage cans, and water supply pipes, and is in utensils, laptops, toys, and many other products. BPA leaches from products into air, food, water, soil, and dust. BPA has links to cancer, thyroid problems, liver problems, and miscarriages, and is suspected to cause effects at extremely low doses. It is estimated that 93% of Americans have it in their bodies due to continual exposure. Researchers, doctors, and consumer advocates want governmental regulation on BPA. The chemical industry insists it is safe, but some countries and states have banned it. There is a full ban in Canada, and it is banned from being in baby bottles in the US and the European Union. Increasingly, companies are voluntarily removing BPA from their products. Next time you go buy a water bottle or canned food, you could easily see a no BPA while you're shopping. Researchers found that most of these products shown here had leached BPA from the container linings. In regards to BPA research, almost without exception, the studies reporting harm received public government funding, whereas those reporting no harm were funded by industry. The take-home point is you should be educated and aware of the chemicals you come in contact with and their potential long and short-term impact on you and your posterity. That concludes the mini lecture on toxicology, dose-response curves, and toxic substances. Please review the weekly outline and complete the activities for this week per the schedule set forth in the outline. Thanks and gig 'em!